Recording is also started. So in today's class, we will look at this paper. This is article number 3. Okay. The title of the paper, as it suggests, that it is studying the role of T lymphocytes, okay, on the wound healing process. So does everyone remember the process of wound healing that we studied previously, or what are the phases? You guys remember, right? Like simple yes and no will work. Yes. Can I assume that you guys recall, means remember the wound healing process, the phases of the wound healing process, and what all cells are involved? Hello, is my screen visible? You guys?
Yes, ma'am.
Yes. So Harshita, do you remember the wound healing process? Don't worry, I won't ask the any question related to
Uh,
it
I'll let you beg.
a little bit. Okay, fine. So I hope at least some fragments of that process you guys remember. Okay, I hope because then only you will be able to understand this paper. That is why I asked. So in let me just quickly recap the wound healing process. So in this paper, I wish to show you this figure. So for in skin, in normal skin, uh, when it is wounded, so the first phase, what happens? The platelets arrive at the wound site and tries to stop the bleeding. That is hemostasis first happens. And during hemostasis, the platelets get trapped in the blood clot or the thrombus that is formed. So the activated platelets then start secreting various kind of cytokines and chemokines to signal the immune cells. Okay? And the inflammation phase begins, where immune cells like macrophages, T cells, and other neutrophils, etc., they arrive. And their job is to fight any foreign agents that might have entered into your skin layer. So this inflammation, it can last for a few days, like 1 to 3 days. It is showing in a timeline. So it is like for hours to minutes, but if it's a large wound, it can go for days. And then once the inflammation stage is over, means you will see there will be reduction in swelling and redness will start going down on the area. Then the proliferation phase begins, where the cells, the keratinocyte cells, the fibroblast cells, the myofibroblast cells of the skin, they all become active. The keratinocytes, they try to reform the epidermis, lost epidermis area through the process called epithelialization. Okay. Then in the underlying layer, the dermis layer, there are fibroblast cells. They try to reconstruct the ECM structure. The blood vessels, new blood vessels starts forming, okay, through the process of angiogenesis. So it happens as the cells are proliferating and signaling each other, okay. So once proliferation is done, you will see that superficially on your skin, the wound is almost like gone. Means that area doesn't ache anymore. Okay. But the healing process is still ongoing because there is the last phase, that is the remodeling phase, that happens over months or years, in which your dermis layer reconstruction continues, but superficially the epidermis layer is healed. And after a few months, you will see some scar forming at that area that shows that the remodeling process has done this. Okay, the scar resolution, that is the end product of your wound healing that happens through during the remodeling phase. So this paper uh, was published in 2019. Okay, in this journal, Wound Healing Society. So if you see the significance, like why should we study wound healing, or why should we focus on tissue repair process? Because and that too in this tissue engineering course. Why? So why do you think a artificial tissue construct would be used? It will be used to either restore, repair, or replace a damaged tissue. Okay. So when your tissue is damaged and you want to repair it, means you're trying to modulate the tissue repair process, then you need to understand the underlying wound healing mechanism that is going on. And you will engineer your tissue construct in such a way that promotes faster wound healing with minimum scarring. That is your ultimate goal. In this paper, they have tried to understand the role of the cell T lymphocytes. Okay, so there are two types of lymphocytes, T and B lymphocytes. And T lymphocytes, they basically mediate adaptive immunity, cell-based adaptive immunity. But the B lymphocytes, what they do, they secrete antibodies. Okay, other than that, there is innate immunity where the macrophages and there's phagocytose, macrophages, neutrophils, the phagocytose. So phagocytosis, they protect our body. So these are two types of immunity. So let's just deep dive in this paper with the simple introduction. So the objective of the paper is to understand the role of lymphocytes, all right, and how they are involved in dermal scarring. So as I said, ultimate goal of any wound healing process that tissue engineers are trying to target is the accelerated wound healing with minimum scarring. So they are in this paper, they are trying to understand how lymphocytes, especially the T lymphocytes, are playing a role in reducing the scarring process. And the scarring process happens in the dermis layer. Okay, so that's why dermal scarring. So they have assessed the temporal spatial distribution of T lymphocytes, means over the time where the T lymphocytes are present during the normal wound healing process in mice. We first understand the distribution of the T lymphocytes and how the distribution is changing over time. And then they compared the scarring outcomes, which means how much scarring. By scarring, what I mean, I'll explain a bit later. So they compared the degree of scarring between the wild type mice and immunodeficient mice, also called skid mice. So the skid mice does not have the lymphocytes in its immune system, but it does have macrophages and leukocytes, WBCs. WBC and macrophages are there in skid mice, but they do not have lymphocytes. So basically, they are trying to compare the presence or absence of T lymphocytes, how it is affecting the dermal scarring process. So what they observed in brief is that this subpopulation of T lymphocytes was present, okay, during so they studied the wound healing process for 30 days. On day zero, the wound was created, and for 30 days, like for 1 month, they saw how the healing process is going on. So here they didn't use any artificial tissue construct to change to accelerate the wound healing process. Here, normal wound healing process is studied that happens without any help of drugs or artificial tissue construct. Okay, so the size of the wound that they must have created must be very small, just like a small cut. Okay, we need large artificial constructs when the size of the wound is very large. The critical sized wounds means Those wounds that cannot heal on itself, it requires assistance. So those for those kind of wounds, they have done this study, and later results we will see as we go on with this paper. So when tissue is injured, it undergoes a complex sequence of repair events, which include the platelet-mediated hemostasis, local lymphocyte activation, and with myeloid cell infiltration, skin cell proliferation, and ECM remodeling, which result in scar formation. Okay, that is what I just said, the four phases. Okay. The inflammation process is an essential phase of the wound healing process, because if the inflammation doesn't happen at all, okay, the inflammation degree of inflammation is very less, or if it is very very large, then both response can lead to excessive scar formation, and thus the tissue integrity will be reduced. So when I say scar, or what is scarring, it means that collagen a large amount of collagen is deposited. Okay. And the cell population in that area is very, very less, almost negligible. Okay. So because of that, that area of that tissue is non-functional. So if there is a large amount of scarring, then you can assume that that particular section of the tissue is not able to contribute to the overall function of the organ. So if the scarring area is very large for a very large wound, then it can also compromise the tissue integrity. It can reduce the uh, hemostatic function of the tissue. So that is why scarring people try to reduce as much as possible through medication and all. But not a lot of success is has been achieved so far. So this kind of research can help uh, us tissue engineers to understand what kind of therapies or treatments we can develop. So let's understand the lymphocytes. Okay, what are lymphocytes? So lymphocytes are from hematopoietic origin, means the hematopoietic stem cells in the bone marrow. They differentiate into the lineage that gives the lymphocytes, and they are very important for immune functions. Now, there are two types of lymphocytes, the B lymphocytes and the T lymphocytes. B lymphocytes account for humoral immunity, means it produces antibodies, and through antibodies, the foreign antigens get recognized and flushed out of the system. Whereas the T lymphocytes, they are cell mediated. So you must have heard antigen presenting cells, and then T cell receptors. So when you were studying immunology, you must be familiar with these terms. So here there are T cells like the killer cytotoxic T cells, which can recognize the foreign microbes that have entered the body and phagocytose them, okay, and then remove from the system. So mainly the cells, different T cell population, they help in protecting us from the foreign agents. Whereas the B lymphocytes, through B lymphocytes, it is the antibody molecules which does the same function. Now within T lymphocytes, there are various subtypes. First subtype is the CD4 T cells. CD4 is the receptor. So T cells which display the CD4 receptor is named here as CD4 T cells. Their role is to release T cell cytokines and modulate immune responses. Right? So their job is to call more and more T cells, okay, at the place. So they're basically the first one to arrive at the wound site and call other T cells, and thus manage how the inflammatory response will go on at the wound site. Then there is another subpopulation, CD8. Now they are cell cytotoxic, means they do phagocytosis to destroy the foreign agents. Also, there are helper T cells. They are expressing a, very unique gene, that is FOXP3 gene. And also there is, and they are also called regulatory T cells. So CD4 T cells, CD8 T cells, and regulatory T cells. So these are few subtypes of the T cells. Now, there are various studies in the literature that have studied the role of B cells, but not T cells. So one study shows that if B cells, B cells, they can alter the diabetic wound healing process, but they have not seen the effect of T cells. So in people with diabetes, their wound healing uh, speed is very less. Means if even a small cut has like developed on their body, it will take a very long time for them to heal, and sometimes that wound becomes chronic. The patient has to continuously eat some medication or uh, continuously dress that wound for years, and still that wound doesn't heal on its own. That is called chronic wound. So one study shows that the B cells plays an important role in accelerating, in making this diabetic wound healing process faster. And so here you can see that here they what they are trying to say is the role of B cells. People have tried to correlate with the wound healing process, but what is the role of T cells and how they affect the scarring process is not very clear so far until this study. So what they are going to do in this paper, they are going to first examine the distribution of the T cell around in the wound area, okay, over 30 days. Then they are going to compare it with the so they are going to do it in the wild type mice where the immune system is intact and all the cell population are present. That is wild type mice, and they are going to compare it with the mice which is lacking the lymphocytes. That is skid mice. Skid mice do not have the lymphocytes, and there they're going to see uh, what happens in absence of lymphocytes. So where lymphocytes are present, they are going to see that in wild type mice. Then what happens in absence of lymphocytes to the wound healing process, and then what they are going to do, they are going to transfer. They're going to transfer subpopulations of T cells from wild type to skid mice and show how the transfer of T cells is changing the wound healing process in the skid mice. Okay. So any doubt so far in understanding anything? Any query? So have you guys understood what they're going to do? Yes,
Yes,
Rashmi.
ma'am.
Okay, so you guys have understood. Okay, thank you. So what so if I were to use these results for my tissue engineering application, means I wish to make such a tissue construct, such a skin construct, which can reduce the scar formation of a chronic wound area. Okay, so I would try to incorporate those factors in my growth factors in my skin tissue construct, which will allow more and more T cells to migrate into that area. Okay, and I can expect that scarring can reduce. So you can also develop some special formulations of T cells containing only T cells, and then use that as medication and see whether it is reducing the scarring process. So this type of paper or research articles where they are trying to understand the underlying processes, what is happening, what is going on. at the cellular level, molecular level, helps a lot while engineering, like tissues and all. That is why this kind of paper are equally relevant for us. So let's see what results did they find. So if any one of you is interested, guys can read the experimental section in detail, um, and ask me later on if you have any doubt in understanding any of the experimental techniques. But the scope of this course is not like helping you understand these techniques. Rather, this paper, the theme of that paper, I want you to understand what is the results they have found. So, which is why I'm skipping the experimental section. But you guys, if you are curious about how did they do it, etc., so you can read. Okay. So first, as I said, they are going to see the distribution of the lymphocytes in the normal wounded area in wild type mice. So at day zero, so this is day zero, and here is the dermis layer, here is the epidermis layer. So this image, then this image, these images are taken through immunofluorescence, means they use antibodies uh, which can conjugate with the receptors on the cell. And these antibodies are themselves are conjugated with some fluorescent dye. And when you observe under fluorescent microscope or using confocal fluorescent microscope, you can observe the cells, where the cells are located. Okay? That is immunofluorescence studies they have done. And they have observed the distribution of CD3 T cells, okay, CD3 T cells, CD4 T cells, then CD8 T cells. So CD8 T cells are the cytotoxic T cells, then CD25 T cells, and FOXP3. So as I said, FOXP3 is the regulatory T cells here. FOXP3 is the regulatory T cells. They have for each subpop. So these are all subtypes or subpopulation of T cells, and they try to see where they are located, okay, in the skin. So for CD3, they observe that in day zero, when the wound is created, these cells are mainly present in the epidermis layer. So this dotted line is the basement membrane of the skin, and the top of the basement membrane is the epidermis. So they are present in the epidermis layer. Now the wound is created at day zero, and they are seeing that in the dermis layer, these are all images for the dermis layer, and the white arrows are showing where the CD3 cells are present. So from this uh, figures, we understand that the CD3 cells are present at all times during the wound healing process till day 30. So this graph also tells you the percentage of cells, CD3 cells. So the black bars, they tell you the cell number over day 1 to day 30, whereas the green line is showing how many CD3 cells are present in the epidermis layer, and these bars are for the dermis layer. So they have quantified it. So these images are a bit difficult for everyone to understand. So which is why this kind of graphs are very much helpful, which quantifies the values. This is a qualitative thing where people observe. Okay, qualitative analysis. This is a quantitative analysis when numbers are given. Quantitative. Now, similar results was observed for CD4 T cells, where the white arrow shows where the CD T cells are present in the epidermis and dermis layer. But but they could not observe CD8 cells, CD20. So they could not observe CD8 cells, CD25 cells, and FOXP3 cells in the dermis. So you can see there are no white arrows for CD8 cells, CD25 cells. So this shows that during the normal wound healing process in a wild type mice, it is the CD3 cells and CD4 cells that are predominantly present throughout the wound healing process, whereas CD8, CD25, and FOXP3 are not that much present. That means they are not contributing to the wound healing process. Okay, this was simple. Then they uh, saw what happens uh, in skid mice when you create a wound. So they monitor the uh, wound healing process in skid mice and analyze how the dermis layer, especially the dermis layer, is getting matured during the healing process. So skid mice, they do not have lymphocytes. So whatever inflammatory response that they have observed is because of the macrophages and the WBCs, the leukocytes. So in this figure, what uh, you will understand is they have compared it. So all the experiments, whenever you do experiments, you always do relative analysis. Okay, you don't do absolute. Means you set up few control experiments, which are positive control or negative controls, and that helps you to analyze where your experimental setup stands. Okay, whether it is supporting your point or it is opposite to what you are proposing. So in the wild type mice, they observed so they observed that the formation of new epidermis, the length, to what area, to what length, not area, to what is the length of newly formed epidermis. So in wild type mice, it is lesser, whereas in the skid mice, the immunodeficient mice without lymphocytes, here the length of newly formed epidermis is larger. So at this point, the it is the boundary of the wound. Okay? This is the yellow arrows is the boundary of the wound, and because of the epithelialization process by the keratinocytes, new epidermis layer is formed. So it has contracted to this much layer length. So it has gone to this much length. So in skid mice, epithelialization process is happening very fast, right? Whereas in the uh, wild type, it is not so. It is not the case. They also checked the scar area. Okay, what is the area of the scar that is formed? So again, here, observe this, the thickness. So in skid mice, it is thicker. The scar area is bigger, whereas in the wild type, it is less. So these images are taken through HNE staining. H stands for hematoxylin and E stands for eosin. So hematoxylin and eosin, they are used to stain both the cells as well as the ECM of the tissue. So the dark purple color, they are the cells. You can think that most cell population is there. Whereas the pinkish regions, slightly pinkish regions, they are the ECM. But these kind of images, I think you might have seen earlier as well in different papers. Then they also studied how much collagen is deposited in the dermis. So during um, scar formation, as I told you, more and more collagen formation takes place. It is also this condition also leads to uh, a medical term that is fibrosis is used for this condition where a large amount of scar tissue or collagen is deposited. It is also called fibrosis. So they use special stains that can only stain collagen. So trichome is used as a stain to understand the level of collagen deposition. So in skid mice, it is the highest. Okay, in skid mice, it is the highest, as well as whereas in wild type mice it is less. And this collagen they form bundles later on. So initially they are just like randomly spread. As the tissue matures, they organize themselves into collagen bundles. So the how much collagen bundle formation has taken place, that is observed through this dye, here we see. So here, in again the skid mice, the collagen bundle formation is the highest. Okay, whereas in wild type mice it is less. And the wound contracture, if you recall, it happens because of the activity of this special type of cells, that is myofibroblast. So this myofibroblast, they are partially. I'm not audible. Am I audible?
Yes, ma'am.
Sorry. Vaishnavi was trying to enter. I mistakenly declined. Can you ask her to join again? Can anyone of you ask her to join again? So apparently to so someone is saying Kirtan. Yes, Kirtan is saying that I'm not audible. Kirtan, can you recheck your audio system? If you are connected to your speaker not properly, because others are saying that I'm audible. So where was I? So I was saying that they also observe the level of the myofibroblast, how much myofibroblast cells are there, because these cells are part muscle and part fibroblast, and due to the muscle nature of these cells, they can contract, okay, and bring the two edges of the wound closer. The wound contracture, they help in wound contraction. So in skid mice, they saw that myofibroblast level is high, whereas in wild type mice it is less. So alpha SMA is a specific cell surface marker for the myofibroblast. So they stain for alpha SMA. So through that they could understand the level of myofibroblast. So this gives us the conclusion that wound repair. Okay, so wound repair, it is happening at an accelerated rate. Okay, means the closure of the wound is happening at an accelerated rate with increased fibrotic scarring in the skid mice, which is not having the T and B lymphocytes. Epithelialization process, you can see the length of the newly formed epidermis is higher. So epithelialization process is more in skid mice. That is, wound closure is accelerated. It is happening fast. It is happening properly. Whereas there is a large deposition of collagen, and activity of the myofibroblasts is also large, means the number of myofibroblasts in this dermis layer is higher in skid in comparison to the wild type mice, because of which there is more fibrosis, means scar tissue formation in the skid mice. So this helps us to understand that in absence of T lymphocytes, because skid mice does not have T or B lymphocytes, this kind of wound repair process will proceed. Now, we also saw the level of inflammation. Okay, how much inflammation is going on in skid mice, as well as whether or not new blood vessels are forming in the wound area. That is through new blood vessels will form through angiogenesis. So in this figure, the CD45 is the marker for leukocytes. Okay, whereas F480 is the marker for macrophages. Okay, means cell surface marker. So anti, so they use antibodies, anti CD45 and anti F480 antibodies to identify the leukocytes and the macrophages in skid mice and wild type mice. And what they observe? They observe that in the SCID mice, the percentage of leukocytes is higher, and as well as the macrophages are also present in higher number in the SCID mice, which means the inflammation process in the uh, wound healing in SCID mice is because of these two cells, and it is happening at a higher level in comparison to the wild type. So in wild type, which is having the lymphocytes as well, there the activity or the percentage of the leukocytes or the macrophages is not that much. So in SCID mice, the entire inflammation process is predominantly controlled by these two cell types. And when they also observe the percentage of capillaries, microcapillaries, okay, at the dermal wounded area in SCID mice and wild type mice. So, blood vessels are made up of endothelial cells, and a marker, cell surface marker of endothelial cell is the CD31. So, they saw that in the wild type, where there is lymphocytes, there the there is more angiogenesis going on, because of which the percentage of microvessels is higher. See, it is higher. Whereas in SCID mice, it is comparatively less. So in SCID mice, they concluded that in absence of lymphocytes, T or B lymphocytes, it is the leukocytes and the macrophages which is controlling the inflammation process, as well as there is not much angiogenesis process going on, because of which there is lesser number of blood vessels formed at the repaired wound area. So these two results they saw at day 7, not day 30. Okay. So day 0 they created the wound, and at the 7th day they studied the level of angiogenesis and the cell populations, inflammatory cell populations. Okay. Now what they did next, they tried to now so far you have understood that in the SCID mice, which is not having any lymphocytes, there the wound repair process is proceeding with the highest scarring, right? Also, not enough blood vessels are getting formed. So, if that tissue area is not getting blood supply properly, then the function of the tissue is compromised of that particular area. So now they want to see whether if we supply T lymphocytes into the SCID mice, whether they can see some change, a positive change, means a reduce in scarring, whether it is possible. So what they will do, they will uh, they will take out some populations of T cells from a wild type mice and then inject it in the SCID mice. So they made 4 groups of, of T cells. So what are these 4 groups? One is the total lymphocytes, means all the lymphocytes, means T cells, B cells, all, and including all subtypes of the lymphocytes. Then in another subset, there is CD8 T cells and B cells. There is no CD4 cells. So this is another subtype, another subgroup that they injected. Next one is there is only T cells of subtype CD3, CD4, and CD25 minus. So they gave short names, okay, to these subgroups. I will briefly mention them here so that you remember them. So there are total lymphocytes. Then there is CD4 minus. So here the B cells are there, and as well as other T cells except CD4. Then there is CD4 plus. So here B cells is not there. and CD4 plus cells is there, and CD3 and CD25. CD3 cell is there, but CD25 minus cell, or sorry, CD25 minus CD25 cells is not there. That is why minus. And next group is TREG, regulated T cells which are having the regulatory function. So here CD3, CD4, CD25, and CD127. So these are the four groups of lymphocytes that they have isolated from wild type mice, which is having the same gender, same age, and same strain as the skid mice. Okay, and then they injected it. And after injection, they assessed the level of these cells. So, and they did it using the technique called flow cytometer. So, in the wild type mice, there are all T cells. The skid mice, there is no T cells. Whereas in one population of skid mice, where they injected total lymphocytes, both B and T lymphocytes, here is the cell number for that, means it is increasing. And this is another subpopulation of T cells, which is devoid of CD4 cells. Okay, so this is the cell number in that skid mice. Here, CD4 cell is there, so it is also showing in that skid mice, and this is the thread group, so it is also there. So through flow cytometer, they confirm the presence of the injected cells in the skid mice. Next, they studied how these injected cells, injected T lymphocytes, is affecting the wound healing process in otherwise skid mice. And they compared it with the negative control, that is your skid, that there is no T lymphocytes. And in that in another group of skid mice, where the T lymphocytes is added, here how the scarring process sector is changing. So see figure B. Okay, this is figure B. Now you will see that the activity of macrophages and the leukocytes, how it is changing in the skid in comparison to the experimental samples. So CD45, that is standing for the leukocytes, the level of leukocyte has actually decreased for total, for the group that is having total lymphocytes, then CD minus and CD4. Whereas in the group which is having the TREG subset of lymphocyte, here the level of activity of CD45 has not decreased that much. It is as comparable to the skid mice. So this shows what the presence of these lymphocytes here, this, and this, presence of these three types of lymphocytes is basically reducing the uh, migration of leukocytes at the wound area, which is why the inflammation mediated by this leukocytes is also less. Then they similarly saw how the activity of the macrophages are affected. So skid mice groups with total lymphocytes and CD4 cells, here only the activity of macrophages have reduced. Whereas in the other group, the reduction is not that significant. Then scarring. So they tried to see what is the effect on scarring. That is how much collagen deposition has taken place. So they saw it through trichome staining. So in this graph, the groups that were having the total lymphocytes and the CD4 cells, okay, and the CD4 cells, here only the collagen content have reduced, means deposition of the collagen have reduced. Again, that means that fibrosis level have declined. In comparison to the other groups, which is the group like CD minus and the regulatory T cells here, the collagen content has not reduced that much. Then the level of activity of the myofibroblast is checked, and they saw that the total lymphocyte population means the skin might having total lymphocytes. Here, the myofibroblast activity has reduced significantly. So there will be a delay in wound contraction. So usually, uh, when there is a delay in wound contraction, it is correlated with reduced scarring. Okay, so most of the wound tissue construct that is made for wound healing application, they try to prove that because their construct is delaying bone contracture, there will be less scarring. So this kind of result is supporting that statement. And also, they saw whether or not the presence of lymphocytes is promoting angiogenesis, means formation of more and more blood vessels at that wound area. And what they saw is the group that is having CD4, there the highest microcapillaries has formed. And then the next is for the group CD minus, and there will be next highest is for CD, the total lymphocytes. So these are the top three groups where the level of formation of the blood capillaries have increased because of the presence of these lymphocytes. Okay, so what did we understand so far with this result? That the T lymphocytes, they are promoting vascularization, means formation of new blood vessels at the area, wound area. Also, there is less deposition of the collagen content, thus the fibrosis level has reduced. Also, the activity of the myofibroblast and the population of the myofibroblast at the wound area, there that has also reduced, because of which there will be less scarring. Okay, so this is possible because the T lymphocytes were introduced, and they say that to these experiments that it is because of the T lymphocytes they could see these results. So in future, if we were to implement this result for our wound healing applications, we would try to uh, either use some growth factor molecules or purposely inject T lymphocytes to achieve this kind of results. So, does anyone have any queries so far? Because I have explained this paper to the best I could, and if you have any queries, I'll be happy to take them. So, have you guys understood what the theme of this paper and what they tried to achieve, and how did they do the experiments that they have done? Have you uh, at least a little bit have you understood? Yes, no.
Yes, ma'am.
So, okay. So I think it was Janvi, right? Who responded. Yeah. So what about others? Is it the same response? Have you guys understood?
Yes, ma'am.
Yes,
Okay.
ma'am.
Okay. So that's good. And if you have any doubts, you guys can let me know. But you should go through this paper once in your free time so that you will understand the theme of this paper better. I chose this paper because I wanted you guys to see what kind of research is going on in this area of wound healing. Because if you see um, in the value of this area and what kind of impact it can have on people through the burn patients and the patients with chronic wounds. For them, if proper treatment can be devised for them, then it will be very, very beneficial for our economy as well as for improving the quality of life for these patients. So, which is why understanding the underlying processes of the wound healing process is very, very important. Then only you will be able to engineer this some kind of medication, not medication, but tissue constructs or uh, some formulation containing cells or some bioactive molecules, which can help the patient to, you know, get faster relief from that wound. So, please go through this paper in your own time and obviously I'll send this recording and you can have a look and then let me take attendance and then we will end today's session. So, hello, am I audible?
Yes,
Hello.
ma'am.
Yes, ma'am.
Okay. So Vaishnavi, are you present here?
Yes, ma'am.
Yeah. Shambhavi, present? Is Shambhavi present here?
Yes, ma'am.
Keaton, don't know if you can respond. Um, Janvi,
Present,
are you
ma'am.
there? Vani,
Yes, ma'am.
Harshita,
Yes, ma'am.
Aditya, Nidhi.
Yes, ma'am.
So I have marked for Vaishnavi, Shambhavi, Janvi, Vani, Harshita, and Nidhi. Anyone else who is left? Ketan, you joined again, no? I think you left the meeting. I'll mark your attendance.
Hello,
Yes,
ma'am.
yes, yes,
Good.
I marked your attendance. So again, I'm repeating. I have marked for Vaishnavi, Shambhavi, Ketan, Janvi, Vani, Harshita, and Nidhi. Okay. So that's it. There's seven of you. So if you have any queries, you guys can let me know. Do you guys have any queries? Otherwise, we can end today's session. Okay. So go through this paper, and again, if you feel any difficulty in understanding anything, you can let me know in coming class. That is on Friday. Okay. So thank you guys for joining. You guys can leave the meeting.
Thank you, ma'am.